Good afternoon and a very warm welcome to everyone for today's online lecture session. We thank you all for joining us today. I am Srinu Appikonda, curator at Birla Industrial and Technological Museum. I'll coordinate today's lecture session. During the sessions, viewers may post their question for the speaker using the chat box, and we will moderate them toward the end of the session. This lecture session is jointly organized by Birla Industrial and Technological Museum and Calcutta Consortium on Human Genetics. With this note, I welcome the President, Calcutta Consortium on Human Genetics, and an Associate Professor, Department of Biochemistry, University of Calcutta, Dr. Saumitra Gupta, to kindly take over the session. Madam, please.
Good afternoon. Honorable Directors, the Director of National Institute of Biomedical Genomics, Professor Saumitra Das, and the Director of Birla Industrial and Technological Museum, Shri V.S. Ramachandran, my revered seniors and colleagues from Calcutta Consortium on Human Genetics, which we lovingly call CC-HULC, and my dear student participants. On behalf of CC-HULC, a warm welcome to everyone. Right from its inception in 2008, CC-HULC, formed and run by scientists and research scholars of different research institutes in our city, has been working relentlessly to promote various research activities and outreach programs to impart education and instill enthusiasm among students about human genetics and genomics. To accelerate this, recently, we have teamed up with the Birla Industrial and Technological Museum, Kolkata, which is also engaged in popularizing and promoting science, especially among the youth. As a part of this collaboration, today's webinar has been organized, and I'm grateful to the Director of BITM for encouraging collaboration with CC-HULC. Thank you, sir. May I now request Shri V.S. Ramachandran, Director, Birla Industrial and Technological Museum, to kindly introduce Professor Saumitra Das, the speaker of this afternoon. Sir.
Yeah, thank you, madam. It is my pleasure to introduce the speaker. Good afternoon, everyone. I am sure that you are all aware that this year's Nobel Prize in Physiology or Medicine has been awarded to Doctors Harvey J. Alter, Michael Hutton, and Charles M. Rice for the seminal research on Hepatitis C virus. Today, we shall learn from Professor Das how their landmark work has contributed to the fight against blood-borne hepatitis, a major global health problem. Professor Saumitra Das, the Director of National Institute of Biomedical Genomics, is an eminent virologist. Professor Das is also a professor in the Department of Microbiology and Cell Biology of the prestigious Indian Institute of Science, Bangalore, where he coordinated the functioning of the Center of Excellence for Research on Hepatitis C Virus. The team led by him was successful in the development of a vaccine for hepatitis C using a mixture of virus-like particles. In addition to making remarkable contribution in understanding the intricate regulations of transcription and translation of hepatitis C virus, Professor Das is also elected fellow of all the major Indian science academies, namely the Indian Academy of Science, the National Academy of Science India, and the Indian National Science Academy, and a recipient of all major national science awards. He is also elected member of several international acclaimed science societies. May I now request Professor Saumitra Das to deliver his lecture on discovery and landmark studies on hepatitis C virus. The forum is open to you. Welcome, sir.
All right. So, am I audible? You can hear me? Can you hear me?
Yes, sir.
Yeah.
Yes, sir.
So, yeah, thanks to the organizers, particularly the Calcutta University, the Center for Human Genetics, the CC-HULC, as Saumitra said, And also, it's a great pleasure to come back to BITM, Birla Industrial and Technological Museum, after 43 years. It so happened in 1977 when I was a student of class 11. I was studying in the St. Xavier's School. At that time, it was actually used to call St. Xavier's Higher Secondary Unit. And I was actually participating in one science seminar, national science seminar organized by BITM. So, and then after I stood first in the Calcutta sector, I was actually representing in the interstate competition. And 43 years back, BITM, actually I remember Bakchi was there, but I don't remember the curator at the time. They imbibed the biology in me. And it's because, and I still remember the topic was important discoveries in genetics. And, I'm just talking about 1977, and genetics was actually a new concept. And with that, actually, when I came back to the our college, I was not having biology. So my, we used to call Father Hink. So he was the principal. He said, how come you are just giving a seminar on genetics and you are not having biology? So that's I think, the turn of my life, and I have taken up biology. And now, almost now reaching 60 years, actually now I'm in an institute where we practice genetics day and night, actually more on the human genomics. So I'm really indebted to the Birla Industrial and Technological Museum that 43 years back, what enthusiasm they have given me, and actually that helped me to carry on my career all the way here. And the school students, this is definitely, I would like to emphasize that this is essentially the Birla Museum have been doing it from my childhood days, as they said that they completed 60 years. And I'm very proud that for the Indian school children and others, they're doing a huge contribution to impress upon into the science, not only in biology, but in the other streams in the multidisciplinary areas. So with this note, now I would like to start my talk. Let me share the screen. So can you see my screen? Can you see my screen?
Yes, sir, it can be seen, sir.
Yes.
All
right. Yes.
All right.
It's all.
Yeah.
Seen.
So I essentially, as I told you, that I have learned that how to impress upon the school children when they are young to the area of science. So that's why I also made it very popular lecture, but some of you might get bored. But anyway, I think you please bear with me. So I'll start with the historical perspective before we really land up with hepatitis C. So the term hepatitis is as essentially thought long back in 3000 BC, where Sumerians, they used to call liver is the home of the soul, and jaundice is the attack on the liver by a devil named Azahu. And in 400 BC, Hippocrates, actually there's a mention that the bile contained in the liver is full of phlegm and blood and erupts. After such an eruption, the patient soon raves, becomes angry, and talks nonsense. Between 17th to 19th century, the jaundice epidemic was very common in the military men and the civilians. In fact, actually, in the civilians, particularly in the French camp, it was actually found that they used to call jaundice de champ, or yellowing of the camps in the battlefield of jaundice. And in the Greek clinicians, they named it as an icteras, which is the yellowish discoloration of the skin. And the myth was that if somebody develops this kind of yellowish discoloration of skin, they should go to the forest and sight the bird, which is the Icteras glabella. And sighting this bird will actually cure them of jaundice. So all these things were actually going on when in 1965, Dr. Bloomberg, he was actually trying to understand the correlation or the genetics of the disease susceptibility among different parts of the world. And he ended up with an antigen, named it as an Australian antigen. This antigen was actually later on identified as the hepatitis B antigen. So in 1965, and then I think Bloomberg got it the Nobel Prize in 1976. So as you know, all of you know, hepatitis can be actually due to alcohol, due to autoimmune disorder. It could be because of non-alcoholic steatohepatitis. It could be because of the toxins and other infections, because of the medications and the drugs. But mostly this is contributed by a range of alphabets: hepatitis A, B, C, D, E. I always call F, could not really sustain, it failed. So hepatitis F you don't see, but then there is hepatitis GB virus. And there are some other viruses can also contribute to viral hepatitis. Those are called non-hepatotropic viruses. Now, if you look at this viral hepatitis and all these A, B, C, D, E, and G, they are mostly all single-stranded RNA virus, except the one that is with the partially double-stranded hepatitis B virus. And you know, hepatitis D also, it's called a viroid. It needs a satellite virus hepatitis B to actually infect and sustain. Now, hepatitis C is essentially was discovered in 1989, and almost similar time in 1991, I landed up in UCLA. I was actually working on poliovirus, and then we switched over to hepatitis C because suddenly NIH funding moved from polio to hepatitis C. Now, in 2007, a study showed that actually hepatitis C, it's actually a silent killer, and it actually infected more than the HIV patients. As you see in that particular area year, it was 15,106 compared to 12,000 for the HIV cases. Now, what is hepatitis? Now, in hepatitis C infection, actually 15 to 25% cases, they can self-resolving. But 75 to 85 cases can actually land up into establishing a chronic hepatitis. And of that, 20 to 30% can cause cirrhotic. And of that, 2 to 7% can end up with the hepatocellular carcinoma or end-stage liver disease. Now, this can be, the hepatitis C can be actually passed on to others by injecting the illicit drug. It could be the high-risk behavior in the past. It could be because of the blood transfusion. Hemodialysis is actually before 2000. All the people were not screened. The blood donors were not screened for hepatitis C. So unfortunately, a huge population got hepatitis C. They don't even know, because sometimes it takes 12 to 17 years to show up. So they are actually carrying the hepatitis C virus, and there was no test available in 2000 to check whether the blood donors are actually carrying this hepatitis C virus or not. So over the years, I mean, since its discovery, people have seen that it is really spreaded all over the world. And as you see, there are several genotypes and several subtypes. There are 7 genotypes and 62, and actually it's keep growing, subtypes. And because of so many genotypes and subtypes, and you know, this is because of the mutations that happened due to RNA-dependent RNA polymerase, which is not very a good student. It copies, but it copies wrong, and that's how it ended up incorporating mutations in the genome. And if the genomes are actually sustained, then it becomes a genotype, and that's how the complications are. Now, coming to the discovery of the hepatitis C virus, 3 people were actually awarded Nobel Prize this year. They are the Harvey J. Alter, Michael Houghton, and Charles M. Rice. Now, what is their contribution and why they were awarded? I would like to somehow touch upon. Now, as I told you that hepatitis, the concept was very old, and hepatitis B was actually discovered in 1965. But people were still observing that there is a virus which is not a hepatitis A virus, not even a hepatitis B virus, but it still causes hepatitis, and people have no clue why it actually, what kind of organism it is. So Harvey Alter, what they did, they have actually taken the serum from a person who is infected from acute and chronic patients and taken that serum and injected into chimpanzee. So when they injected the chimpanzee, and the chimpanzee developed into a viral hepatitis. So that was the clue that it actually ends up, and remember, this serum was already screened for hepatitis B. That means this serum did not have hepatitis B or hepatitis A, but still it could cause hepatitis in chimpanzee. And that's how the identification, or actually the first observation by Harvey Alter, who showed that the chronic infection of this is non-A, non-B viral hepatitis is actually, it is sometimes more frequent than hepatitis B. It was named as non-A, non-B until then. And this non-A, non-B could transmit the disease to chimpanzee. And another thing they just found, that the virus is actually somehow coated with the lipid. So that means somehow it actually needs lipid, or it uses lipid for its propagation. So these are the things, and its size was actually 30 to 60 nanometer that was reported by Harvey Alter. But then actually Michael Houghton came, and this is around 1970, when he actually showed that this is not 70, it's actually much later than that. So it's actually, they showed that this infectious agent, if you actually isolate the serum, pellet it, isolate the DNA and RNA, and then make a shotgun library in lambda GT11. So this is essentially a library, a cDNA library, where you can express the proteins. And that expressed proteins were probed with the antibody collected from a patient who has non-A, non-B viral hepatitis. So you have the serum collected from a patient with non-A, non-B, isolated DNA and RNA, made a library, made an expression library so that the proteins are expressed. They were actually immobilized in a plate, and then they were probed with the antibodies collected from a non-A, non-B patient, and this keep on screening. After a lot of screen, they ended up with a clone, which is called the clone 511. And these, they actually found this is the one that causes the non-A, non-B hepatitis. And you can actually see the original Western that actually was published in the science paper. And it showed that this is the non-A, non-B hepatitis is only lighting up, not with the hepatitis B, not with the hepatitis A. So then Charlie Rice came into the picture. As I said, that people were actually, soon after the discovery, they were interested to work on it and then try to see whether they can actually clone this hepatitis C virus. So Charlie made an effort to isolate the RNA from the infected patient. And in that RNA, somehow he made the cDNA and found that actually there is a conserved non-coding region at the 3' end. And he is the only one who pointed out that the presence of that 3' end non-coding region is very important for the replication of the virus. So when he engineered this RNA with this 3' conserved region, that RNA was infectious. And if you inject that RNA into chimpanzee, you can make it with the liposomes or some other delivery vehicle, it can come up with a hepatitis C infection. So this was a landmark discovery, I will say it, because that is first time we could reproduce the infection from a viral RNA. So, and Charlie showed that this infection, now if you take out the serum from chimpanzee, isolate the RNA and infect it into a cell culture, and he was actually using human hepatocellular carcinoma, or HUH7. Actually, he made it HUH7.5 to be more precise. and showed that this particular infectious RNA, if you just add to this cell culture by the liposomes or some other vehicle, this can replicate in a cell culture. Why it is so important? Because we already know that this is a virus, and this virus is an RNA virus characterization and everything was common. But cell culture is very important so that actually we can go ahead with the research and understand the biology of the virus. So to actually summarize this part, so all it was a discovery of a non-A, non-B viral hepatitis. From the blood transfusion, the person gets it, and from the serum isolated from that person, if you can inject it into chimpanzee, you can see that the chimpanzee comes back with the disease, and then the viral RNA isolated from there, we could show that the hepatitis C antibodies are actually there. And Charlie Rice showed that when you clone this RNA and then make a cDNA, and that cDNA you transcribe to make in vitro transcribed RNA is good enough to come back with the infection in chimpanzee and also can replicate in a cell culture model. So this landmark discovery by these three gentlemen actually showed now how this hepatitis C virus can be handled. So now detection was easy. So people come up, now the cell culture is possible, so people could actually grow the virus. They can make the antibody in mice or in rabbit, and then actually make the proteins first, and then actually they can show that you can come up with a detection kit for detections of the hepatitis C virus in the blood donors or any suspected individuals. Not only that, it eliminates the risk of transfusion of the associated hepatitis, and there are effective antiviral drugs. As you know, now there is drug in the market like tenofovir and sofosbuvir, which can actually successfully reduce the virus titer and cure the patient. So the disease can now be cured and raises the potential for a global eradication of the hepatitis C virus infection. So with this is the discovery. Now I will just come to a little bit of biology of the hepatitis C virus to impress upon that how their discovery helped others to understand the biology of the virus. Now this is an RNA virus. As you know, the virus goes inside the liver cells, uncoats and makes the viral RNA. That RNA is then used to make proteins, viral proteins, and those viral proteins are, some of them are required to bind to the viral RNA and replicate from the positive strand to the negative strand. And the negative strand is then used more and more as a template to make more and more of the positive strand, because the positive strand is the one that will be packaged inside the virion, which we call virion assembly, and then finally releases out and then infects the neighboring virus. So what is interesting is all these hepatitis virus somehow select liver as their target. And hepatitis C particularly is actually more selective. They select the hepatocytes within the liver and not the neighboring cells such as the stellate cells or the Kupffer cells. Now, it is something intriguing because hepatocytes are occupying almost 80% of the cells, but there are endothelial cells, there are stellate cells, there are Kupffer cells, they are spared. So I can actually visualize something like an intruder gets into a house and they exactly know where to go within the house, to go straight to the kitchen and make use of all the vessels within the kitchen to make palatable food. Because they know that if they land up with another room, they might be caught by the people, or they won't be actually able to make their food because they need to make food to sustain within that system. And that's precisely happens for the hepatitis C. It goes into the liver cells, goes into the hepatocytes and makes a very, very palatable sub-sandwich. And this is exactly like the polyprotein of the hepatitis C viral protein. and that is cut like we cut it with a knife with small pieces into structural and the non-structural proteins. There are structural proteins like core, envelope, E1, E2, then there are P7, which is an ion channel protein, and there are non-structural proteins. Particularly of interest is the NS3, which is a protease, and you know that this is the protease which is targeted for designing antiviral. There is NS5B, which is RNA-dependent RNA polymerase, and there is NS5A that is also actually targeted for making drugs. So before the discovery of the telaprovir, sofosbuvir, and ledipasvir, actually people used to use pegylated interferon or ribavirin, but they were actually not so effective. And later on, people found that the telaprovir, sofosbuvir, and ledipasvir are actually more efficient in bringing down the virus titer. But any drug will always have some problem with the specificity, and particularly for the hepatitis C, there is so many genotypes. I told you 7 genotypes and 62 subtypes. So it is very important that you keep on looking for alternative, because there are chances of not being successful to eradicate the virus, and there are emergence of the drug resistance. There are mutants which actually keep growing, and there are additionally therapy associated limitations with the drug. So people keep trying to see whether we can have better and better drug to really eradicate this virus from the world. Now I'll just highlight a little bit of our work, what we have been doing. Now in our lab, we have been working on protein synthesis. Now, as we know that in general, the cellular proteins, they have a mRNA which is capped at the 5' end. There are initiation factors binds, and that attracts the 40S ribosome with the tRNA, looks for the initiator AUG, 60S joins, and then translate and makes the protein. But strangely, in the hepatitis C virus, there is no 5' cap structure at the end of the viral RNA. Instead, there is a structural RNA which forms a scaffold of structure, and that attracts the 40S ribosome to bind on near the initiator AUG. And that helps. This is called internal ribosome entry site, because it binds to an internal sequence, not to the 5' end of the cellular RNA. And we showed actually now this internal initiation helps to bypass the cap-dependent translation of the cellular RNA. Not only that, while we were actually initiating this work in 1998 in my lab in Indian Institute of Science, there was a paper in Science which actually Jennifer Doudna, the other Nobel laureate for the CRISPR-Cas, she showed along with Jerry Frank by cryo-electron microscopy that hepatitis C actually binds to the exit gate of the ribosome. In the ribosome 40S, there is a head, platform, and the beak. The A sites and the P sites are in the front, but the hepatitis C somehow binds to the exit gate, and it avoids the competition of the front runners in the A sites and the P sites. Now, after it binds, it actually triggers a conformational alteration and access to the A sites and P sites so that it can easily translate the protein without competing with the viral RNA. Since this was unique and fundamentally different, we wanted to understand which all ribosomal proteins are actually involved into these interactions of the viral RNA with the ribosome, which all host factors which are involved into this interaction, and which are viral proteins which regulates this process of translation. And over the years, we have been working on it, and we are trying, since it is unique and fundamentally different, is it possible that we can target it to design selective antiviral? And you know, we have been reading that there are so many antibiotics which actually affects the ribosome and the bacterial RNA translation, interaction and translation inhibition. So here we have proposed that if you use an RNA, small RNA, which can block the exit gate of the ribosome, which can prevent the viral RNA interaction with the ribosome, then we can achieve the translation inhibition. In fact, one of my first students, he showed that this small RNA can block the viral RNA interaction with the ribosome, and we can win the race by inhibiting the viral protein synthesis. And since it is binding to the exit gate of the ribosome, it doesn't really interfere with the cellular RNA translation. So now this whole process is actually mediated by several proteins. It's not so easy. There are several proteins which are actually nuclear protein. They come out from the nucleus to the cytoplasm and then helps in the virus to translate this whole thing. So, and we have shown that this protein comes out, their affinity changes towards the viral RNA. So they forget about their cellular function and they started like a hijacking conditions helping the virus. So that's how it has been going on. So I actually told my students, it looks like a tug of war between the host cell and the virus. There were several proteins we have shown. One of the proteins we recently showed, that HUR protein, which helps the virus to actually tilt the tug of war. And then there is a microRNA which prevents that. It looks like that there is something which can still work on it. And this is one more protein which we call human LA autoantigen or LA protein, which is something like a Ola on the roads, which can always tilt the story on your favor. But again, all these things are actually under the traffic light. And I consider that the traffic light is nothing but the phosphorylations or PTM. It's like a post-translational modifications of the proteins, which decides the affinity of these proteins, whether to bind with the viral RNA or to bind with the cellular RNA, whether to help the virus or whether they should continue helping the host. And that's essentially we are trying to understand. So overall, it's like an orchestra, and hepatitis C is actually tuning this orchestra, and everybody is dancing with that tune. There are proteins like HUR protein, PTB protein, LA protein. They binds to the 5' UTR or 3' UTR. I'm not getting into the details. And there are microRNA and long non-coding RNA, which actually helps in this orchestra. And the whole thing somehow manages how the virus can successfully translate their RNA, avoiding the competition, establish chronic infection within the liver cells. So over these years, we have been showing that what all the viral proteins, microRNA, or the long non-coding RNA that are involved into this. As I told you that we showed LA, PTB, HUR, which has nuclear proteins and also helping in viral RNA translation replication. We talked about the microRNA 122, microRNA 125B, and there is a long non-coding RNA very recently we have showed, which is called HULC, and this is heavily expressed in the liver cells. Actually, in collaborations with Dr. Soma Banerjee's lab in IPG-MER, we showed in the tissues also this HULC is highly upregulated in the hepatitis C infected patient. We did some RNA sequencing as well, and we showed that this HULC is very important, and it is specifically upregulated in the cirrhotic liver or in the hepatocellular carcinoma liver. And then we tried to understand the biology, how a long non-coding RNA. Remember, people used to write it off that long non-coding RNA does not have any function. But over the years, we have learned that long non-coding RNA actually could be the game changer. The small one, the microRNA, and the long one, the long non-coding RNA. The non-coding RNA could actually be the game changers. And we showed that how the HULC gene is actually modulating the viral infection, and also in terms, HULC is regulated by the microRNA 122. And I'll just show you with a cartoon that how hepatitis C virus actually plays the game within the liver cells. This is a game between your Tom and Jerry or Minnie, and you can see how the spike can also come and then help this and ultimately win the race, because what happens, the HULC regulates the viral infection. This is a proviral. At the same time, HULC is being regulated by there are microRNAs and other proteins which sequesters HULC, and HULC again helps the virus to propagate from one cell to the other cell. a single long non-coding RNA can do triple function to actually establish chronic infection in the cells. So this is something which is very important. I'm just showing you the cartoon because I just believe that when people do research, they should visualize actually what is going to happen. If you don't visualize, if you don't know why you are doing, then it's very difficult to motivate ones. Now, all these experiments and all these leads that we obtained was very difficult to prove it in an in vivo situation because we don't have any mice model for hepatitis C. So we collaborated with a laboratory in Japan who has humanized mice. So these mice actually harbors human hepatocytes, and since they are harboring human hepatocytes, they can be infected with the hepatitis C virus. In that system, we have actually tried our antivirals. And as you see over the years, although we started the lab with a simple translation of the viral RNA and ended up doing a whole bunch of translational work and showed that how the small RNA, as I told you, can inhibit the viral RNA interaction with the ribosome. I talked about a protein called La protein, and we showed that if you just take RNA binding motif of the La protein, make a small peptide. This we did it with Dr. Siddhartha Roy at the IIC. At that time, he was in IICB Kolkata. We solved the structure, enema structure of this beta hairpin structure of this peptide. We showed that if you add 6 arginine residues, this peptide can go inside the liver cells and inhibits, like a surgical strike, it can inhibit the hepatitis C in the liver cells. We showed some other peptide like an NSC protease. This is a viral protein that also binds to viral RNA and regulates its translation replication switch. And if you use a peptide corresponding to the RNA binding motif, this peptide prevents the viral protein to bind on and inhibits the viral proliferation. Government of India asked us to look for herbal compounds. So we used actually pomegranate and Phyllanthus amaranth. These are the two things which are known to be hepatoprotective, and we isolated the pure compound from them, and we showed that how they can be effectively inhibiting the viral protease or the RNA-dependent RNA polymerase. And this actually we are trying to commercialize and see whether we can have a effective antiviral which can be used as an add-on therapy along with the drugs that are already in the market. Because of the good thing is, now the drugs, as I told you, they are good in reducing the virus titer, but often the patient progresses to hepatocellular carcinoma. So we are trying to see this add-on therapy would help to prevent the progression of the disease to hepatocellular carcinoma. Along with the decrease in the virus titer, if you take these things, you can actually ensure that the patient will be completely cured. There is very little chance that it will progresses into further cirrhosis or the liver hepatocellular carcinoma. As it was mentioned by in the beginning by your director, that we have also worked with a VLP-based vaccine. VLP is actually a virus-like particle where you have the structural protein. It doesn't have the nucleic acid inside, so it is non-infectious. So we showed that if you make a virus-like particle in a baculovirus system in an insect cells, this is good enough to elicit the sufficient immune response. And this we showed it in mice, and now we are trying to do it in a pig. But remember, they are not infected by hepatitis C, so we just try to see whether they are immunogenic or not. And the next step is essentially we try to see whether this can be actually taken into preclinical trial to human trial. And all these things we are actually testing it in the mice system, where actually the humanized mice system, where we are testing the efficacy of this antiviral, whether they are actually really good enough and expected to work in the human trial. And all this work, one thing I would like to say, that all of you, the future budding scientists, this is the message that I would like to share with you, essentially that the collaboration is the key. You can't do everything this time on your own. So you need to stretch out and reach out to the people who can be helpful to get your dream come true. So, and that's basically our lab have been working on antivirals, As you see, I discussed about the RNA, the peptides, the small molecules, and the structure-guided chemical biology. We also work on the biology of the viruses like host-virus interaction. Shubhendra is working with us in IICB Kolkata. We work with the microRNA and host proteins, and we are also working on long non-coding RNA with Shoma Banerjee in the pathogenesis sector. We also collaborate with Shiv Sharin at ILBS, Rakesh Agarwal, SGPGI. Now he has moved it to JIPMAR. We work with liver mechanobiology. This is something new, as I told you, that in the future, this is all a multidisciplinary work. You can't really address any question with a single discipline, with a single collaborator, with a single person. You need to stretch out and try to see whether you can have a multidisciplinary approach to address a single question in depth. The research question is always simple, but you should actually address it in depth so that the answers are actually strong and you have the real proof of concept. And we have tried with the liver mechanobiology. In fact, one of my PhD students, I can proudly say, is from mechanical engineering. And all he showed that how hepatitis C virus infected liver cells changes the mechanical properties of the liver cells, and how we can have a biomarker to predict whether this liver cells is likely to progress into cirrhotic liver or fibrotic liver, or may not really get into those complications. We also work with the liver targeted different drug delivery, work with the virosomes with Devi Sarkar's lab at ISAR. Mohali, he was at the Delhi University at the time. Now he's back in Delhi University. We work with dendrimers, where actually this is with the organic chemistry, and also with the physicist Ajay Shud and Prabal Maiti. And we also work with the nanocapsules with Ashok Raichur in the material science. VLP and DNA-based vaccine, I told you that we have done. This was started with an Indo-Australian project, but now we are actually trying to do it at the peak with the help of in my laboratory at the Indian Institute of Science, and with a commercial lab called Shikra Lab. They will be doing the VLP work in a GLP manner and try to do the preclinical studies. In addition, we have collaborated with Anjali Karande's lab and Soma and Shaila's, Professor Shaila, where we showed that we have isolated the neutralizing antibody from patient sera and showed that neutralizing antibody, if you now convert it into monoclonal antibody, that monoclonal antibody can be used as a therapeutic approach to actually cure the patients. So all these things are possible to address one particular virus. And that's precisely, I'm just trying to say it, that although we feel that the hepatitis C virus is solved, if you really talk to a clinician, particularly gastroenterologist, they will say, we don't have to worry about hepatitis C anymore. There are 99% cases their virus titer goes down in 3 weeks time. But what we feel that the question still remains. We still don't have an appropriate model for this organism. Chimpanzee is the one, but that is heavily discouraged. People are trying mice model, but they are not naturally susceptible. You need to make it humanized, which is not very cost effective. Or people try to use the ectopic liver generations to use that model. Vaccine is still not in place. We talk about COVID vaccine, but this is now more than 32 years old, but we still don't have a vaccine for this particular virus. It is difficult because of this the high sequence error prone replication of the NS5B and extensive glycosylations of this envelope protein. And also we can't really make large amount of this virus. Unlike any other virus, this virus does not grow it in cell culture robust way. So we can't really make tons of this virus. And finally, we could not really answer how to stop the progression into hepatocellular carcinoma. This is a really major problem. The virus titer goes down, but the patient actually progresses into hepatocellular carcinoma because the pathways that are activated needs to be identified, needs to be stopped so that this progression can be prevented. There are epigenetic modulations in the RNA binding protein and the small RNA non-coding and coding. We are trying to identify how these epigenetic modulations are actually contributing to the disease severity. And finally, there are a lot of secretory molecules, particularly the exosomes, which are actually contributing to the disease progression to the other organs. We're trying to understand how these things are possible. So there are plenty of things to solve for a particular organism before we can see that the problem is solved. And I was actually congratulating Charlie after he got the Nobel. And he also said that let's hope that this is an example of the benefit that science can bring and will encourage the public to embrace and support fundamental and curiosity-driven research. As we are in the midst of the experiencing with the ongoing pandemic, there are many challenges ahead. So essentially, what I would emphasize to all these youngsters is actually you try to remain curious and ask curiosity-driven questions and try to answer using collaborations and using multidisciplinary approach. Success will be coming on your way, and this is a burning example. I have seen Charlie and collaborated closely with him, so I know him that if you really stay focused, you can reach the goal. So I would say, arise, awake, and stop not till the goal is reached. And with that, I would like to conclude. Thank you all. This is my present lab. As you know, my major lab is at the Indian Institute of Science, but in NIBMG also we have now recruited 3 postdocs who started working on hepatitis C virus and COVID research, and we will continue contributing in the field of research and then actually, as I told you, to answer our curiosity-driven questions. Thank you all. Thank you.
Thank you very much, sir. Indeed, it is a comprehensive and elaborative session. Our young students put up some of the questions. May I take, sir?
Yeah, sure.
Biswas Pandit wants to know, is there any particular drug that has been found or manufactured that can be used against all genotypes of the virus?
No, there is no pan genotype as such, but there are tenofovir and boceprevir. This tenofovir and boceprevir is actually working more or less with majority of the genotypes and subtypes.
Yeah,
but we can't really claim them as a pan genotype.
Yeah.
People are trying, but there are claims like Lady Pasvir, people are saying pan genotype, but now people saying some of the subtypes, the response is not so good. Any other question?
Another student wants to know. Riddhi Sen Gupta wants to know. Sir, could you explain the difference between acute and chronic, and acute chronic, and that is with respect to HCV infection?
So when the virus is actually detectable and actually it shows its phenotype, we call it an acute disease, because you can easily probe them, you can detect them, you can treat them. So, or we call fulminant. But when this is actually goes into hideout and it's we feel that we actually got rid of the virus and it actually goes to the hideout and remains there for long time, then it becomes a chronic. Now for hepatitis C virus, it has been shown that in the beginning, it becomes a, either it is challenged by the immune system and flushed out. So that is the self-resolving the disease part, which I said 15 to 25% resolves it. But the remaining part where they maintain a chronicity, that chronic virus is very difficult to detect and then actually eliminate. So it is very important to eliminate this chronic virus because until it shows up, and in India we don't have a practice of testing every year whether you got some infections or not. I mean, after the COVID, people are actually more alert about blood tests and other things. Earlier, I mean, a large population doesn't even go to the clinician to check whether they're having any routine tests necessary or not. So it's very difficult to detect the chronic viruses.
Yes, sir. Next. Sana would like to know how long a person live after is being diagnosed with hepatitis C. C does
No,
mean like, 1% detected there.
no,
Like,
this is very relative question because it's all depends. And liver, even if you take out half of the liver, people can live. And there are cases even a complete liver can be transplanted. So I won't say that there is something like a danger signal or a time period. So it all depends on genetic determinant, predisposition, and other comorbidity. All things determines whether the patient will live longer. But many cases the patient actually battles for a long time, and there are now a lot of treatment, supportive treatment, which can take care and then keep the patient alive for a long time.
yes, sir. Now, next, Kaushik Mitra wants to know, are there any approved vaccines now or over the horizon for the HCV, as there are vaccines for hepatitis A and hepatitis B?
Say it again, please.
So he wants to know, like, there are vaccines for hepatitis A and B. Similarly, that the vaccine is available for HCV. I mean, C,
No,
hepatitis.
hepatitis C, there is no vaccine available so far. People have been trying it, but none of them are actually gone into phase 3 so far.
Okay. Gargi Datta wants to know, how can we overcome the problem of hepatitis C transmitting during the window period among the blood donors?
So it's essentially the testing. One has to test the donors for hepatitis C. So then you can eliminate.
Yes. Swapnamay Chakrabarti, next question is from him. What is the window and incubation period of hepatitis C? If a disease is somehow be detected in window, I think similar kind of question. I think you answered it already.
Actually, so the window is 12 to 17 years. It can be as late as 17 years.
Okay, 17.
They may not even know that they are carrying the virus.
Yes, sir. Yes, sir. Sir, next, another question. We know the liver can reaction so that can mass and protein. One more question, sir. Saugata Kumar Pal. We know that liver can regenerate, so can't we treat it with RNase enzyme to treat the infection?
RNase enzyme.
Yes, sir.
We can do it in vitro, not an in vivo situation.
That is thing, sir, and questions are there, but have time, sir. A couple of things like, which may be we come across this lot of questions by our students, like this I think happens to be positive stand RNA virus, and SARS-CoV-2 as well, positive sense RNA virus. So do positive sense RNA virus have advantage over other viruses, means to cause in terms of like virulence?
So it's not that. I think if I reframe the question, see, the main problem with the RNA viruses is the faulty enzyme of RNA-dependent RNA polymerase. The RNA-dependent RNA polymerase actually have a tendency of incorporating wrong bases. And since it is faulty and they don't have the proofreading, so that's how there is mutations. And if the mutations in a hotspots or a wrong pocket, then it can alter the epitopes and the immunogenicity. So it may not be, it can bypass the antiviral, it can bypass the antibody response, everything can be bypassed. So that's how we are actually fighting so much with the RNA viruses. In general. That is common between COVID and hepatitis C or chikungunya, anything if you think about.
Yes, that is it. So similarly, you have mentioned your one of the slides that, like, this causative of the hepatitis disease are so many factors like genetic, metabolic, or autoimmune. Like, from these all these factors, actually autoimmune very common here. autoimmune with this hepatitis, actually what it does, sir, for the students to understand actually how.
Hepatitis is can be caused by autoimmune disorder, not C. Hepatitis C is different. Hepatitis C is a virus which
Yes, complete
induces
way.
hepatitis. And what I talked about, that is hepatitis in general. Anything goes wrong with liver, we call hepatitis, right?
Yes.
Anything wrong with the hepatic liver, so is actually called hepatitis. So that is actually meant by the autoimmune disorder and the effect on the hepatitis. But hepatitis C and autoimmune disorder, I don't think there is as such any connection established yet.
Yes. Yes, sir. Madam, would like to.
So, if there are any questions, you have any questions queued up?
Yes, madam. Questions are cleared. No question. I think.
So, I have a question, Professor Das, for you. Is there any known loci, I mean, like human genes or human variants that has been known to be associated with hepatitis C mediated disease susceptibility?
You know, there are a lot of work on IL-28B.
Okay.
People used to say that that can say the susceptibility, but they became later on couldn't really sustain. I mean, if you really ask Sridhar Chinnasamy, he will say lambda 4 is one of them. Now the thing is, essentially, there is no such strong correlations established so far. People have been working and then they are trying to see there is a disease susceptibility. People used to call there is mutations in not in actually in the virus, the ISDR and PPHD region. If you have a mutation, then they are not susceptible to interferon, and then actually they can be easily challenged, those kind of things. But now it looks like there are a lot of other things that we don't know. You know, to be precise, I think it's more on microRNA and the long non-coding RNA, that and their crosstalk with the virus is more important for the
genetics.
For the yes. And actually, you are right. As I mentioned in the last slide, the epigenetic changes in those RNA, epigenetic changes and the modifications in the RNA, it could be viral RNA, it could be cellular RNA, microRNA, or long non-coding RNA, is actually the key, which we never really looked at it. Now people are trying to find out whether that can be correlated with the genetic determinant. That can be the genetic determinant. Epigenetic.
Thank you.
Yeah.
So, do we have time in hand?
Time means it is. We have come to the conclusion. I think questions are not seen.
Before we end, I would like to thank Professor Saumitra Das for giving this wonderful talk in a very short notice, because I know that I have just probably 8 days back I sent you the letter and you graciously accepted our. So on behalf of CCU, I thank you very much. Thank you so much.
Thank you, sir, on behalf of the BITM. And despite of having busy schedule, and you spared some time for us and coming, addressing our viewers. And we are thankful to you and grateful to you. And our director is with us and would like to.
Let's have a formal thanks for all your efforts. We are very much impressed with your talks. Okay.
Thank you. And then on behalf of NIBMG, I can tell you that at any time, if you need help or you want to send students, you are most welcome. We'll be happy to. I mean, once this COVID situation is over, we can start entertaining people, because we also have a good museum done. Pathoda has made one human genetic museum. I don't know whether you have seen it or not. So we want to make it more public, but we could not do it because of the COVID. We are just waiting these COVID things to get over by this year, hopefully.
Sir, I was very much happy. I was very much happy when you went down to your memory of visiting BITM, and I was so happy. Because a lot of high-level people always talk great things about BITM. That is a great strength for us.
Actually, I was telling my daughter before coming to this seminar that actually, I'm feeling very proud that after 43 years, I am going back to BITM. Thank you.
Thank you.
Indeed, we are happy, sir, to hear that.
Sure.
Thank you so much.
Looking forward. See you, sir. Namaste. Namaste, madam.
Thank you, madam.
Thank you.
Thank you.
So the meeting is over.